Cell division is fundamental to life, but not all cell divisions are the same. Two critical processes, mitosis and meiosis, serve very different purposes in living organisms. This diagram shows the key differences between these two processes. Mitosis on the left produces two identical cells, while meiosis on the right creates four genetically different cells through two divisions. Mitosis is the process used by somatic cells, the regular body cells that make up your skin, muscles, organs, and most other tissues. Its primary purpose is growth, repair, and maintenance of the organism. Meiosis, in contrast, is a specialized process that occurs only in germ cells to produce gametes, sperm and egg cells. Its purpose is sexual reproduction and creating genetic diversity. The fundamental difference lies in the cell types involved. Mitosis works with somatic cells, your everyday body cells. Meiosis works with germ cells to create specialized reproductive cells called gametes. Another crucial difference is chromosome number. Mitosis maintains the diploid chromosome number, meaning cells have two copies of each chromosome. Meiosis reduces this to haploid, where cells have only one copy of each chromosome. In summary, mitosis is your body's way of growing and repairing itself by creating identical copies of cells. Meiosis is nature's way of ensuring genetic diversity by creating unique reproductive cells that combine during sexual reproduction to form new organisms. We'll now examine the detailed steps of both mitosis and meiosis, comparing how these two essential cell division processes differ in their stages and outcomes. Mitosis consists of 6 main stages that result in 2 genetically identical daughter cells. Let's examine each stage in detail. During interphase, the cell grows and replicates its DNA. In prophase, chromosomes condense and the spindle apparatus forms. Metaphase sees chromosomes align at the cell's center. Anaphase separates sister chromatids to opposite poles. Telophase reforms nuclear envelopes, and cytokinesis physically divides the cytoplasm, creating 2 identical diploid cells. Meiosis is more complex, involving 2 sequential divisions. Meiosis 1 reduces chromosome number, while meiosis 2 separates sister chromatids. Meiosis 1 includes prophase 1, metaphase 1, anaphase 1, and telophase 1. This is followed by meiosis 2 with similar stages but different chromosome behavior. Prophase 1 of meiosis contains the most crucial difference from mitosis. Homologous chromosomes pair up to form structures called tetrads through a process called synapsis. During crossing over, non-sister chromatids exchange genetic material. This process creates new combinations of genes, dramatically increasing genetic diversity in the resulting gametes. The pattern of chromosome separation differs significantly between mitosis and meiosis, leading to different outcomes in chromosome number and genetic composition. In mitosis, sister chromatids separate, maintaining the same chromosome number. In meiosis 1, homologous chromosomes separate while sister chromatids stay together, reducing chromosome number by half. The end results clearly demonstrate the fundamental differences between these two processes. Mitosis produces identical cells for growth and repair, while meiosis creates diverse gametes for reproduction. These step-by-step differences ensure that mitosis maintains genetic consistency for body cell functions, while meiosis generates the genetic variation essential for species survival and evolution.